This is Pratima from Planet Physiology. Today we shall have Introduction to Renal Physiology, where we shall discuss physiological anatomy and functions of kidneys. Renal system includes kidneys, ureter and urinary bladder. Humans have a pair of kidneys located retroperitoneally, that is posteriorly behind the peritoneum on the either side of vertebral column. Usually right kidney is slightly inferior to the left due to its relationship to the liver. Weight of kidney is about 150 grams in male and 135 grams in female. Each kidney is surrounded by a fibrous capsule and is embedded in perirenal fat. Suprarenal glands are located on the superior aspect of the kidneys. Each kidney has a medial indentation through which renal artery enters the kidney and renal vein and ureter leaves the kidney. So shown in this figure, this is hilum showing renal artery, renal vein and ureter. Internally, kidney is divided into outer cortex and inner medulla. Same is shown in this longitudinal section of kidney. Renal medulla consists of striated conical renal pyramids with their bases at periphery. Apices of renal pyramids form renal papillae. They drain into funnel-shaped opening of the renal pelvis called minor calyces. 2 or 3 minor calyces fuse to form major calyces, which in turn form renal pelvis. It continues to form ureter and leaves the kidney. Renal cortex is the area between renal capsule and the bases of renal pyramids. Renal cortex also runs between the pyramids as renal columns. Cortex mainly contains major part of nephrons, the Bowman's capsule, proximal convoluted tubule and distal convoluted tubules, while the loop of Henle's and collecting ducts are present in medulla. Shown here is one enlarged nephron with its distribution in cortex and medulla. As the renal tubules run parallel to each other in the medulla, they give striated appearance to the renal pyramids. Now let us study the structure of nephron in detail. Nephron is basic structural and functional unit of the kidneys. There are about 1 million nephrons in each kidney. The first part of the nephron is a blind, double-walled, expanded structure called as Bowman's capsule. It has tuft of capillary network within it, which is called as glomerulus. Both the glomerulus and the Bowman's capsule together is called as renal corpuscle. Other end of Bowman's capsule gives rise to renal tubule. The first part of the renal tubule is proximal convoluted tubule. As name suggests, it is a coiled or folded structure. It becomes straight and continues in medulla as thin descending limb of loop of Henle, which then takes a U-turn and runs towards cortex again as ascending limb of loop of Henle. Initial part of ascending limb is thin and hence known as thin ascending limb, and the later part is thick and hence called as thick ascending limb of loop of Henle. As it enters the cortex, it forms distal convoluted tubule. It runs between afferent and efferent arterioles of the parent glomerulus and then becomes convoluted to form distal convoluted tubule. Now the tubule once again becomes straight to form connecting tubule. Many connecting tubules join to the cortical collecting ducts, which again enter the medulla and form medullary collecting ducts. The medullary collecting ducts finally form papillary ducts, which open into the papilla. Now let us study the important features of various parts of nephron. These features make them specialized for performing specific functions. Let us begin with the Bowman's capsule. As I mentioned earlier, it is a double-walled, cup-shaped structure. Its parietal wall is lined by simple squamous epithelium, but the visceral wall has specialized epithelial cells called as podocytes. They wrap themselves around glomerular capillaries, which are highly fenestrated. Same are shown in this picture where podocytes are labeled as P. As seen in this diagrammatic representation, podocytes have many finger-like processes which surround the capillary walls. Narrow gaps between the adjacent processes are called as slit pores or filtration slits. These filtration slits are bridged by a thin diaphragm called as filtration slit diaphragm. Slit diaphragms are actually specialized form of tight junctions. Key structural proteins of these diaphragms are transmembrane protein called nephrin and Neph1. They are present on the tips of podocytes and form meshwork and also possess strong negative charge. Thus, they restrict filtration of proteins. Mutations of nephrin gene is associated with congenital nephrotic syndrome of Finnish type, which is associated with massive proteinuria. All these features help glomeruli in selective filtration of plasma. Mesangium is the third component of renal corpuscle. Mesangial cells are derived from monocytes and are present close to the glomerular capillaries. They provide structural support to the glomerular capillaries and they also secrete extracellular matrix or mesangial matrix, prostaglandins and cytokines. These cells are contractile in nature and hence regulate surface area available for the filtration. As these cells are phagocytic cells, they also remove foreign organisms and debris in the kidney. Mesangial cells that are present outside the glomerulus are called as extraglomerular mesangial cells or Lasix cells. They are also known as gurmatic cells. They secrete small quantities of renin and erythropoietin and are also phagocytic in nature. The gap between parietal and the visceral wall is called Bowman's space, which is continuous with the proximal convoluted tubule. Filtrate enters the lumen of PCT from Bowman's space. Now coming to the proximal convoluted tubules or PCT. PCT is present in the renal cortex. Its initial part is highly coiled and hence called as pars convoluta, while the lower part, which is straight, is called as pars recta. There are some functional differences in the types of channels present in these two parts. PCT is lined by cuboidal epithelium and it possesses extensive microvilli on the luminal surface forming brush border. It increases surface area available for reabsorption by 20 times. Brush border is also loaded with various carrier proteins. Cells also possess many mitochondria indicating their high metabolic activities. The basal surface is also folded to form labyrinth and usually mitochondria are present in this labyrinth. Epithelial cells possess tight junctions but these are leaky to water and other solutes permitting paracellular transport. In fact, entire renal tubular epithelium has tight junctions, but the main component of the tight junctions, claudin, varies in different segments and hence the type of claudin determines the leakiness as well as selectivity of tight junctions for paracellular reabsorption. Their details we shall study in the tubular function video. PCT reabsorbs most of the filtrate. Now, as PCT enters the medulla, it continues as thin descending segment of loop of Findlay. Epithelial cells become squamous and also lack brush border. They possess few mitochondria indicating their low metabolic activities and mainly the passive role in reabsorption. Then as this descending limb takes U-turn, it continues as thin ascending limb of loop of Findlay for some distance and then becomes thick ascending limb. Thin ascending limb of loop of Findlay may or may not be present in the nephron. It depends on the type of nephrons which we shall study later in this part. Thick ascending segment possesses cuboidal epithelial cells with smaller number of microvilli and moderate number of mitochondria. This signifies their active role in reabsorption of solutes. Now, as thick ascending segment enters renal cortex, it forms another segment of convoluted tubule called as distal convoluted tubule or DCT. Initial part of DCT passes between afferent and efferent arterioles of the same glomerulus, where the cells of both DCT as well as arteriolar smooth muscles are modified to form a specialized structure called as Juxtaglomerular Apparatus. The meaning of juxta is close. So this structure is located close to the glomerulus, so it is known as Juxtaglomerular Apparatus. In this region, the epithelial cells of DCT become columnar and are densely packed. Hence, they are known as macula densa. They act as chemoreceptors and detect sodium chloride concentration in the filtrate that enters the DCT. Smooth muscle cells of afferent as well as efferent arterioles are also modified to form juxtaglomerular cells or JG cells. These are enlarged and granular in nature, as you can see here, and they synthesize and store inactive form of renin, that is prorenin. Apart from macula densa and JG cells, lassis cells or extraglomerular mesangial cells also are the part of JG apparatus. These are agranular cells and fill the space between afferent and efferent arterioles as well as DCT. As mentioned earlier, they secrete small quantities of renin and erythropoietin. JG apparatus in general plays important role in regulation of GFR, that is glomerular filtration rate, as well as renal blood flow. After JG apparatus, the tubule forms distal convoluted tubule. It has two functionally distinct subsets, early and late distal convoluted tubule, which are also known as DCT1 and DCT2 respectively. Epithelial cells are cuboidal in nature. Their luminal surface has few microvilli, but basolateral surface has extensive foldings or labyrinths. They also possess more mitochondria, indicating their role in active reabsorption. This segment is permeable to solutes, but impermeable to water and urea. It can absorb water only in presence of antidiuretic hormone. So please remember, normally DCT is impermeable to water but is capable of absorbing water if ADH is present. Late distal tubules then forms connecting tubule that connects to collecting ducts. Many DCT empty into collecting ducts. Histological features and hence the functions of late distal convoluted tubules and collecting ducts are similar. Both these segments contain cuboidal type of epithelial cells which gradually increase in height to form columnar cells. And these cells are of two types: pale staining principal cells and dark staining intercalated cells. Principal cells are also called as P cells. They are concerned with sodium reabsorption, which is regulated by aldosterone. Principal cells can also reabsorb water, but only in presence of ADH. The second type of cells are intercalated cells, which are also known as I cells or dark cells due to their darker staining properties. These are mainly concerned with primary active secretion of hydrogen ions and reabsorption of bicarbonate ions. Hence, they play important role in acidification of urine. As cortical collecting ducts enter the medulla, they are called as medullary collecting ducts. Similar to cortical collecting ducts, these cells also have fewer mitochondria and are permeable to water only in presence of ADH. They also secrete hydrogen ions by primary active transport process, but this segment allows diffusion of urea also. So the important difference between cortical collecting ducts and the medullary collecting ducts is permeability to urea. Medullary ducts are permeable to urea, but cortical collecting ducts do not permit diffusion of urea. They are impermeable to urea. Medullary collecting ducts are the final site of urine formation, and hence The final urine volume and osmolarity is decided by the processing of filtrate in the medullary collecting ducts. This was the basic structure and their functional relevance to the parts of nephron. Now let us study types of nephron and differences between them. There are two types of nephrons, cortical and juxtamedullary nephrons. As name suggests, Bowman's capsule of cortical nephrons lies in outer cortex, whereas that of juxtamedullary nephrons are located in deeper cortex close to the junction of cortex and medulla, and hence these nephrons are called as juxtamedullary nephrons. About 85% of all the nephrons in the kidney are cortical nephrons, while 15% are juxtamedullary nephrons. Cortical nephrons have short loop of Henle's which run very short distance in the medulla. They also lack thin ascending limb of loop of Henle. Juxtamedullary nephrons have long loop of Henle which penetrate the medulla almost to the papilla. They also have long thin ascending limb of loop of Henle. Usually glomeruli of cortical nephrons are smaller and their filtration rate is also less than that of juxtamedullary nephrons. Tubules of cortical nephrons including loop of Henle are supplied by peritubular capillaries, but in case of juxtamedullary nephrons, loop of Henle's are supplied by special capillaries called as vasa recta. Main function of cortical nephrons is urine formation and excretion of water-soluble waste products, but juxtamedullary nephrons mainly are concerned with concentrated urine formation. Now let us have a look at innervation of kidney. Kidneys are innervated by sympathetic nerves which mainly innervate afferent and efferent arterioles, vasa recta, glomeruli, cortical tubules and JG cells. Sympathetic stimulation causes decrease in renal blood flow and hence GFR due to its predominant action on afferent arterioles. It also causes increase in renin secretion which plays important role in maintaining GFR and blood pressure. Kidneys are not innervated by parasympathetic nerves. Now let us throw some light on functions of kidneys. Grossly, we can divide functions as excretory functions and non-excretory functions of kidney. The first excretory function of kidney is excretion of water-soluble metabolic end products of the body like urea, uric acid, creatinine, bilirubin and end products of hormone degradation. Apart from these, kidneys also excrete foreign substances that enter the body through various routes like drugs, pesticides and chemicals ingested along with the food. Non-excretory functions are the functions of kidney other than excretion of various substances. These functions include regulatory functions, endocrine functions and gluconeogenesis. Let us study them one by one, starting with regulatory functions. The first one is regulation of water and electrolyte balance in the body. The same is also referred as regulation of extracellular fluid volume and osmolarity. Since regulation of water and various electrolytes composition in the body go hand in hand, this function is considered together as a single function, regulation of water and electrolyte balance. By regulating excretion of various electrolytes like sodium, potassium, chlorides, bicarbonates, magnesium and phosphate, kidneys help in maintaining extracellular fluid osmolarity. As these are osmotically active particles, it also affects water reabsorption and hence the fluid volume also is regulated. The hormones which act on the renal tubules include aldosterone, angiotensin, atrial natriuretic peptide, antidiuretic hormone and parathormone. Regulation of water and electrolyte balance helps in maintaining normal cell volume and hence their functioning as well. The next regulatory function is regulation of blood pressure. Kidneys play important role in both short-term as well as long-term regulation of blood pressure. JG cells of kidney release renin in response to decrease in blood pressure. It converts angiotensinogen to angiotensin 1 and then to angiotensin 2. Details of renin-angiotensin system is covered in separate video. The link is provided in the description box below. Vasoconstriction by angiotensin II to increase blood pressure is part of short-term regulation, while release of aldosterone by angiotensin II and then its effect to regulate blood volume is part of long-term regulation of blood pressure. Kidneys also excrete excess of water and sodium by the processes called as pressure diuresis and pressure natriuresis. These processes reduce excessive blood volume and hence regulate blood pressure, and this is also the part of long-term regulation of blood pressure. Kidneys also play important role in regulation of acid-base balance or maintaining pH of the body fluids. They excrete either excess of hydrogen ions or hydroxyl ions as per the requirement of the body to maintain normal pH of the body fluids, which is 7.4. Renal tubular cells also have ammonia and phosphate buffer system which help to excrete required quantities of hydrogen ions without much decrease in urine pH. 90% of the total erythropoietin is secreted from interstitial cells of peritubular capillaries, and hence anemia is common feature of renal diseases, and hence kidneys are the major regulator of erythropoiesis. And therefore, regulation of erythropoiesis is also one of the regulatory functions of the kidney. Next is role of kidney in regulation of serum calcium level. Active form of vitamin D, which is 1,25-dihydroxycholecalciferol, is synthesized in kidney. It helps to increase absorption of calcium from gastrointestinal tract as well as reabsorption of calcium and phosphate from renal tubules to maintain serum calcium levels. Now coming to the endocrine functions of kidney. Kidneys secrete various hormones like erythropoietin, renin, 1,25-dihydroxycholecalciferol, prostaglandins, kinins and thromboxane A2, and thus they act as endocrine organs also. And the last non-excretory function of the kidney is their role in gluconeogenesis. Their capacity of glucose production is almost same as that of liver, but this function is seen only during prolonged fasting, that is during prolonged starvation. So these were the functions of kidneys, and that's all for this session. Thank you. If you enjoy my sessions, press the like button and share it with your friends. If you haven't yet subscribed my channel, press the subscribe button. To get notifications about new releases, press bell icon. Thank you for watching and see you in the next video.